Well, it's a pleasure to be here. What I'd like to persuade you is that systems approach to disease is going to be vital to drug discovery. And I think there are, in my view, 4 elements that are really critical for drug discovery. 1, does the patient have a disease? 2, if the patient has breast cancer, can you stratify that disease into its different subtypes so you can, in principle, have a proper impedance match against drugs? And I think stratification of disease is one of the critical features that is going to really transform medicine in the future. 3, what drug targets do you use against a given subtype of a disease? And I would argue just looking at signal transduction pathways is a narrow and totally incomplete approach to drug target discovery. And of course, the question of making the drug. How do you make drugs that have less cross-reactivity, that are stable, that can be taken orally, all of the ideal kinds of things? And I would argue that the systems approach I'll be talking about really speaks to each of these 4 issues. So systems medicine is the systems biology approach to disease. And I would say it has 2 major features. Disease, like biology, is all about complexity, and the question is how we deal with the enormous biological complexity. And of course, one way that we can deal with it is my prediction in a 5 to 10 year period. Every single patient will have a virtual cloud of billions of data points, and we'll have the computational wherewithal to reduce that data dimensionality to simple hypotheses about optimizing wellness and minimizing disease for each individual. And I would say there are 2 grand challenges in this view. One is the enormous question of how we integrate multi-scale heterogeneous data together to give models that are actionable and predictive. And that is indeed an enormous challenge. And the second that I think a lot fewer biologists appreciate is the idea in any very big data set, there's an enormous amount of noise. How do you extract small signals from enormous amounts of noise? And it's here I think the systems approaches are absolutely critical. A second feature of systems medicine is the idea that one way you can transform enormous amounts of data into useful insights is to create what we call the network of networks. You know that biological networks in principle operate at the chromosomal, the protein, the cellular, at the organ level, and even at the level of individuals. And they are, in an individual, seamlessly integrated to deal with the information they must. In disease, that network of networks becomes disease perturbed, and that alters the information that that disease perturbed network can express. If you can capture that altered information, and systems biology gives us approaches for doing so, you then have new insights into mechanisms of disease as well as new approaches to diagnosis and to therapy. Now again, I really want to emphasize that the systems approach to data and dealing with complexity really has 4 components. 1, all analyses should be as global as possible. You never know which elements are informational elements are relevant to disease. That it is possible to be global with genomic analyses, and much less so with the other omic analyses. The dynamics of systems, both temporal and spatial, are really key to dealing with signal-to-noise issues, and I'll illustrate that in just a moment. And we need to be able, as I said, to integrate these different levels of data together to create models that are predictive and actionable. And again, I'll just emphasize there are really 2 types of noise that you have to be worried about. One is the technical noise that comes from making the measurements and dealing with them in conventional ways. And the other is any aspect of phenotype you assay is fed into by many types of biology most of which are irrelevant to the disease biology you're interested in. How do you subtract away the normal biology? And I'll give you in a moment an example of how we do that. I think there are 5 major conceptual pillars for dealing with complexity in systems medicine, and I'm going to focus on talking about the last 3, the holistic dynamical approach to disease, emerging technology and system strategies, and finally some of the analytic tools that are going to really open up our ability to identify drug targets really effectively. So let me tell you very briefly about the dynamics of neurodegeneration in mice induced by infectious prion particles injected into the brain. And the basic experimental protocol was to look at the progression of this disease, which occurs over 22 weeks, at 10 different time points, quantifying the transcriptome in disease-infected animals and subtracting from that their normal littermate brain transcriptomes. And the idea was to identify the genes that are differentially expressed in disease and normal. And of course, we were horrified to learn that 1/3 of the mouse genes were expressed in the disease brain, differentially expressed. And that meant there was an enormous amount of noise. And most of it was biological noise. And what we did to deal with the biological noise was to construct 6 inbred prion strains of mice, each of which was designed to be able to subtract away from the diseased animal different aspects of biology. And when we did so, we reduced the 7,400 differentially expressed genes to about 300, and these, we felt, encoded the prion neurodegenerative response. We looked dynamically at the histopathology and identified 4 major types of networks: prion replication, accumulation, glial activation, and 2 forms of degeneration. What we did then was to take the 300 DEGs that we felt were associated with neurodegeneration and map them dynamically across the 10 time points into protein-protein interaction networks, which reflected each of these 4 different processes. And we came to a number of striking conclusions. The first of which was the idea that the 4 networks became disease-perturbed in a sequential manner, starting with the most unique network, prion replication and accumulation. And this is really important to disease, because if you're thinking about therapy and/or diagnosis, you really want to know where the disease starts, what the nature of the disease is about, and from a therapeutic point of view, can we perturb this proximal disease-perturbed network and eliminate the disease perturbation of the downstream networks. What was surprising is when we mapped the 300 differentially expressed genes into the 4 major networks I mentioned, it turned out that 200 genes mapped in, but the other 100 actually defined 6 additional networks that changed dynamically. And these were networks that people had no idea were associated with the disease. And the really important point is when you collectively looked at the dynamics of those 10 disease-perturbed networks, you could explain virtually every aspect of the pathophysiology of disease, and you began to get an idea of possible targets for therapy in the future. And I'll just tell you, we've looked at 6 additional mouse models from this dynamical point of view, and in all cases we've found that we've come to similar kinds of conclusions, and I won't go on and say any more about that. But rather, let me move to emerging technologies and system strategies that open up our ability to explore new dimensions of patient data space. Novel and emerging technologies, we have incubating at ISB an absolutely fascinating 3rd-generation sequencing company. And by that, I mean a company that uses single molecule sequencing, nanopores or nanochannels, and electronic signal detection. And what was interesting about this company is, one, by putting two different forms of plastics together, this was a spin out of Intel, they can actually create nanopores whose size is carefully designed, and you can infinitely parallelize the right nanopore sets. And number two, they developed a strategy of being able to park a nuclease right on the lip of each of the nanopores so that you could capture a single strand of DNA and then clip off the nucleotides one at a time and send them through the nanopore to be interrogated electronically. And the advantages of this are, one, that nucleases are infinitely processive, so you can think about 200 kb reads. That means de novo assembly. And number two, sending the individual bases through individually allows you to interrogate their electronic configuration in such a way that we'll almost certainly be able to look at the 16 different epigenetic marks that covalent modifications of DNA. So you'll be able to read out at least the DNA part of epigenetics. The nucleases work for RNA as well as DNA, so you can sequence either type. And of course, the ability to parallelize these things means that in principle, you could think about sequencing the human genome in 15 minutes. And my prediction is in a 5 to 8 year period, if funding is forthcoming for these third generation sequencing companies, and that's a challenge, I'll tell you, that we'll be able to have a human genome at around $100 or so. The second technology I'll just describe is a new form of mass spectrometry called SWATH that has the ability to make mass spectrometry much more global or comprehensive in its analysis. So you can readily envision in complex mixtures being able to identify 6,000 or 7,000 different proteins and to look dynamically at samples and see how they change, just as we now can look dynamically at transcriptomes and microbiomes and so forth. The other technologies I'll discuss later in my talk, so I won't say any more about those. But I do want to talk about four strategies for systems medicine. One, a systems-driven approach to blood diagnostics, which I think is really going to make blood a window into distinguishing health from disease. And I'll argue now we can make a diagnostic for any disease that you have good phenotypic assays for. Number two, we're using induced pluripotential stem cells and following their development to differentiated cell types to look both at normal development and the development that ensues in individuals with disease genes that perturb those developmental processes. Number three, I'll just briefly mention some of the system strategies for disease stratification. And finally, we'll talk about a microfluidic peptide protein capture approach to being able to assay wellness using a droplet of blood. So, systems diagnosis and making a window into distinguishing health from disease. As many of you know, the IOM published a report on the use of omics for diagnostic markers, and they made three really salient observations in 2012. One, virtually all of the preceding diagnostic markers that had been done never were transferred to useful, effective drugs. And number two, the reason for this basically was everyone used the algorithm of look at the blood of normals, look at the blood of diseased, you see lots of differences. But the challenge is 99% of the differences are noise. So how do you sort out the signal that's relevant to diagnosis? And the third really important point they made is that you can do diagnostics within the context of a given population and overfit it beautifully and get high sensitivity and specificity, But if you look at other populations, you can see those diagnostics don't apply at all. So in the future, what is critical for diagnostics is front end to do multiple populations simultaneously, and that's one of the really key ways of sorting out signal from noise. So our belief is blood is this wonderful window. It bathes all the organs. All organs secrete proteins and other molecules into the blood that reflect the status of the organ. And if you have a way of identifying where those molecules come from, you can do blood diagnostics. And I'll talk later about organ-specific blood proteins that enable us to do that beautifully. And in fact, I won't talk about cell type-specific blood proteins, but they also are going to be very powerful for organs that have very complex physiologies like the brain and like the kidney. Longitudinal analyses are key. Multi-parameter panels are absolutely critical, and quantitation is really key. The approach we've taken to blood diagnostics for proteins has been targeted proteomics, where in an hour or so you can analyze quantitatively at the mid-atomole level roughly 100 proteins or so. And Rob Moritz at ISB recently has developed panels of targeted proteomics or SRM proteomic assays for virtually all of the human proteins and frankly all the mouse proteins too. So we have catalogs now where you can take candidate proteins and get the diagnostic reagents you need to interrogate complex mixtures like the blood. What we have also done is analyze the transcriptomes of 50 organs from humans and roughly 50 from mice, comparing each organ against all other organs, asking how many organ-specific transcripts are there. And we find for most organs there are hundreds of organ-specific transcripts. And we've looked and studied 3 in particular, and that's the brain, the liver, and more recently the heart. And what we see is really interesting. So these organ-specific proteins then can reflect changes that occur in the brain, say. So if you have a normal brain, the level of those 200 proteins will be set at one level. If it becomes disease-perturbed, the disease-perturbed networks whose cognate proteins are in the blood will have their concentrations altered. And since every disease alters differing combinations of disease-perturbed networks, you, for each disease, get unique blood protein fingerprints. And we've shown all of this in various mouse models for neurodegenerative disease, for example. But what is really striking is most of the 200 proteins then map back to fundamental physiologic functions in the brain. So not only can you use pattern recognition to distinguish health from disease, if disease which, you can begin to make inferences about what functions might be perturbed as well. And we used 15 organ-specific, brain-specific blood proteins in the prion case, where those 15 proteins mapped evenly into the 4 major networks we saw. And we made 2 really striking observations across the 22 weeks of analysis. One, we could do extremely early detection. We could see changes in the blood of the first of the disease-perturbed networks 10 weeks before any clinical signs whatsoever. And number 2, we could successively follow the disease perturbation of each of the 4 networks so we can follow progression. And I'll give you an example of where we've taken a similar kind of approach with some differences to look at a diagnostic protein panel that can distinguish neoplastic from benign lung cancer nodules. This was done with a company that I started about 5 years ago, Integrated Diagnostics, and Nathan Price at ISB. And of course, the dilemma for the pulmonary oncologist is in the US, there are 3 million cases of lung nodules a year, and the question is, what do you do with these people? And of course, at the extreme ends, you can have watchful waiting, or you can go immediately to surgery. But there are other of the order of 600,000 patients a year that fall in this middle. We're not sure quite what to do. And there you can do PET scanning. You can look for pulmonary aspiration, or you can do bronchoscopy. All expensive, morbidity-causing procedures. And the real dilemma is 40 to 50% of the patients on which these procedures are carried out, and that includes the surgery too, turn out to have benign nodules causing enormous cost and enormous morbidity. So the approach that we took was to use a variety of systems approaches to identify almost 400 candidate proteins for lung cancer. I won't go through the details, but they were straightforward. We were able to detect routinely in the blood of all normals and all diseased about half of these, 190. The second thing we did was to match in appropriate ways benign nodules, patients with benign nodules and neoplastic nodules, and then look at their plasma, analyzing all 190 of these proteins, and then rank ordering them according to the number of accurate 144 calls that they could make, getting the right benign and the right neoplastic. And then what we did was took the top 32 of those proteins, and we created 1 million panels of 10 and rank ordered those 1 million panels with regard to their scoring ability. And then we asked which proteins were most cooperative. That is, which proteins were found most frequently in the highest and most effective ranking panels that we saw. And it turned out that 13 proteins scored very high in this regard. And those 13 proteins did not rank among the first 13 proteins in the 190 score. And in a sense, that's a beautiful systems approach to looking at proteins that interact with one another rather than at absolute rank scores. We then did a validation study with new sites and new benign and neoplastic nodules, and the validation study turned out even better than the original discovery study did. We've done a second validation study, and it turned out very well as well. And we're now commercializing this. And I'll tell you, it's really been quite spectacular in terms of having people rush to this. We now have several large insurance companies that have agreed to insure and pay for this, and we're approaching coverage now of 100 million patients in a matter of about 2 months, which I think is pretty striking. But one of the interesting questions that you can, and this has been published in an article in Science Translational Medicine in October. But what we can do then is we have a classifier that is 92% effective in calling malignant lung cancers, but what's really great about it is it's 40 to 50% effective in identifying benign patients and putting them in a rule-out category in the watchful waiting. And if you do the calculations, you'll save north of $3.5 billion a year in the US for this, as well as giving a lot of patients peace of mind. So here's the rule-out box with the 144 patients we looked at, and you can begin to see that we will have a few neoplastic there that will have to be watched, but there are a large number of benign ones. And if you, so that's the impedance match between benign and neoplastic. But if you look in the real population, you see the vast majority are benign, and again, the rule-out works really well in the 50 or even 60% level. So what is striking is if you ask what biology is reflected in those 13 highly selected proteins, and what we can show is 12 out of 13 map into one of 3 disease-perturbed networks that operate in small cell lung cancer. So there is a beautiful biology reflected in this neoplastic process. And I think one of the really interesting questions is, can we use the diagnostics to really move toward choosing effectively therapeutic targets for creating drugs for. So these panels will have 6 features. We'll distinguish normal from the diseased. We'll be able to do early diagnosis. We can follow progression of disease and consequently the response to therapy. We'll be able to stratify disease into different subgroups for impedance match against effective drugs. And finally, we can begin to study for the first time from the blood the dynamics of human disease. And my view, next generation strategy for diagnostics will be, are you well or are you sick? If you're sick, what are the organs that are affected? If a given organ is affected, what is the nature of the disease? And what is the stage of the disease? And finally, we'll stratify that disease into its different subtypes for appropriate impedance match. Let me say, and I'll say something about this again later, in almost all diseases, multiple organs are affected. And what is fascinating about that is you can think about the therapeutic and diagnostic implications of that statement. And of course, the stratification is something I'll talk about yet again in just a few moments. But let me move to the second of the strategies, using induced pluripotential cells for biology and diagnostic, a collaboration with Cellular Dynamics International, who spent $100 million learning how to industrialize this whole stem cell developmental process. And that gives us enormous advantages over the do-it-at-home types that are trying to study development. And the real key, I think, is going to be single-cell analysis, because I think in all populations of cells, you'll find quantized cell states. And here's an example of something we did about 5 or 6 years ago, where we looked at a glioblastoma cell line, quantified 32 transcripts at that time, and were able to divide those 32 cells into 3 discrete quantized groups, quite separate from one another by the quantitative expression of state-specifying transcripts. And I won't get into that too far. But the important point is there are several possibilities for these quantized cell states. One, they represent successive stages of a developmental process, in this case, some kind of neoplastic process. Or 2, they are states of cells that interact with one another to do important things. And we can distinguish between those 2 possibilities once we've thought about quantized cell states. And without getting into a complicated discussion, let me just say that you can map quantized cell states beautifully onto dynamical landscapes illustrated in this central picture here. And you can begin then to think about what are the effectors of transition or effectors of interactions. And you can actually do mathematical modeling for all of this that is very, very powerful. But let me give you some specifics. So what we've decided to do is, with collaborative cellular dynamics, look at the differentiation, a 30-day period or so, differentiation and selection, to neurons and to cardiomyocytes. And the key idea in quantized cell states is most of the transcripts, when you quantify them, are noise. How do you identify the transcripts that are state specifiers? And I'll just say transcription factors, signal transduction molecules, some cell surface molecules are all candidates for state specifiers. So we looked at, for example, the differentiation of iPS to cardiomyocytes, and we could show beautifully at 3 different stages there were whole sets of different transcription factors that one could see. So the approach that we're taking is to, across this 30-day episode, start with 1 billion cells, so we have plenty to do cell sorting, do at 8 different time points single-cell analysis to identify quantized cell states, identify cell surface molecules, and sort these cell states into populations that can be then interrogated with global omic analyses, and I've listed those here. And I'll give you an idea in a moment of the initial start of this whole thing. We also plan to use iPS cells from individuals that have genetic defects that modify these developmental processes. And we're going to look at GATA4, modifies cardiomyocyte development, and Huntington's disease that modifies neurons. And here's the 5-generation family with a GATA4 defect. And we'll be looking at 3 individuals, 4, 5, and 6, from that most distal family, a normal male and 2 males that have 2 different phenotypes of this cardiovascular myopathy and so forth. And the question there, of course, is how does the disease gene perturb the developmental process? Here on the x-axis is the analysis of 96 different transcripts, and on the y-axis is the analysis of individual cell types. And you can see if we analyze fully differentiated neurons and the iPS cells from which they're derived, that you can use the single cell analysis to distinctly identify these 2 different populations. And in fact, if we focus on the developmental neurons, you can see that they actually divide into 4 distinct populations. The majority are the classic GABA or glutamatergic neurons, but there's a smaller population that are poised, ready to go to either of the major neurons, and an even smaller stem cell population that still exists here. And if you do a principal component analysis, you can separate the populations quite nicely. So the idea then is to be able to interrogate all of those quantized populations with enormous omic data and then make computational models that can follow the entire developmental process, both as a consequence of normal development and as a consequence of disease development. Stratification is really key for successful disease treatment. And just to give you a simple example, if you look at blood diseases, 60 years ago, there was just a cancer of the blood. And 50 years ago, it was leukemia or lymphoma. And 40 years ago, there were 5 different possibilities. And today, this is a late slide. I have no idea how many different possibilities there are. But there's been an enormous increase in the survival rate as we've succeeded in this stratification. And my argument would be we have to carry out similar kinds of stratification for all of the major diseases. So one way we can stratify is getting iPS cells from diseased individuals, placing those differentiated cell types in a test tube, and then interrogating them with appropriate environmental stimuli so that we can identify the nature of the disease-perturbed networks. Each stratification will have a different combination of disease-perturbed networks. And we're beginning to do this for several diseases now. We can use, as I showed in the case of prion disease, dynamic networks to be able to stratify disease beautifully. We can use the organ-specific blood proteins, and I'm going to say more about those in just a few minutes. And there are other blood diagnostic approaches, the lung cancer example that I've given you here. And finally, we can do family genome sequencing, integrating genetics and genomics in a beautiful way to enable the identification of disease genes and the definition of disease types beautifully. And again, we don't have time to talk about that. But let me talk now about the final topic, a futuristic approach to wellness using microfluidics, peptide protein capture agents, and organ-specific blood proteins. We now have 19 major organs in the human body covered in the sense that we have blood-specific proteins for each of those organs, so we can quantify the state of those organs across the longitudinal period of time. And this opens up the possibility of doing enormous dynamic human physiology development, physiology, aging, wellness, disease diagnostics, drug toxicity, multi-organ responses to disease. I'll mention that in a moment. So what I'm going to talk about now is a wellness assay where from a drop of blood, we may be able to assess 50 different human organs. And the idea, of course, is to have 50 organ-specific blood proteins for each year, 50 major organs, to create ELISA assays for each of those, and then a microfluidic chip that would be able to quantify each of those 2,500 blood proteins. And the work that has been done here is really pioneered by Jim Heath and his colleagues at Caltech. He has, in fact, developed a microfluidic protein chip that can do 50 ELISA assays from a fraction of a droplet of blood in 5 minutes. And this has actually been put at UCLA to look at drug responses of neoplastic patients to various therapies. But the real challenge here, of course, is if you're to have 2,500 different ELISA assays, it would be prohibitively, if not impossible, to imagine that coming from antibodies. So again, Jim and his colleagues have been looking at peptide protein capture agents, 5 or 6 D-amino acid libraries they've constructed. They've used click chemistry, which puts together monomeric forms of these things in the appropriate three-dimensional orientation. And more recently, they've discovered that circular peptides have higher sensitivity and affinity than their linear polymers. Jim and I started integrated diagnostics 5 years ago. They worked on this peptide protein capture agent approach, and more recently we've spun out another company called Indie Molecular, which is focused just on the peptide protein capture agents. So the idea here is that you can create a library of reagents that have alkenes and a library that have N3 molecules, and you can link those active couplers with different lengths of carbon bonds so that you can use the protein target to juxtapose 2 monomers in exactly the right orientation for click chemistry to couple them together to give the right three-dimensional orientation to create a dimer, or more recently, Jim has created tetramers. And what's really exciting is once you've done that and you know the right coupling lengths and everything, you can, with carpocatalysis in vitro, actually make the coupling reagents. You don't need the protein to catalyze it. And we just want to say that the proteins are really critical because they are used to create the agents that are going to be really effective with regard to sensitivity and with regard to specificity. These protein peptide capture agents are incredibly stable, and here's an example of how their affinity was absolutely unchanged after 2 months of storage at 25 to 57 degrees. So you'll be able to put this in an envelope and ship it to Africa, and its affinity will be absolutely unchanged. And more recently, what Jim has demonstrated is the precise targeting of peptide epitopes. And what this does is really cuts down on the cross-reactivity of these protein capture agents, which is really a critical point both for diagnostics and for therapy. Jim and his colleagues have made about 20 of these protein capture agents, and they've been used for in vitro diagnosis, in vivo diagnosis, and you can actually show that some of the protein capture agents directed against epitopes can block the function of proteins, so they have the potential, obviously, of being drugs as well. So I think this opens up an enormously exciting opportunity. These reagents are stable, they're highly sensitive. Roughly with each mer you add to them, you can increase a log or so in sensitivity. And I'll say in that regard, the circular peptides are significantly superior to the linear peptides. And in fact, dimers of the circular peptides approach specificity, sensitivity of the best monoclonal antibodies. It's digital in the sense Once you know the coupling agents and their lengths, you can synthesize these in unlimited quantities. You don't need to worry about mice and bunnies. And of course, if they can be precisely directed at epitopes, I think you can avoid a lot of the cross-reactivity problems that not only plague antibodies, but plague the small molecule drug compounds as well. We're in the new company going to adapt it to large-scale production. So we see in vitro, in vivo diagnosis and the possibility of these being therapeutic reagents in the future is very exciting. And I think if all goes as we think it's going to go, there's a good possibility these will replace monoclonal antibodies in a 10 to 15 year period. Okay, the final thing I'll talk about then is some of the computational tools that we've developed to begin thinking about the whole drug discovery, drug issue. ISB has about 1/3 of its faculty involved in computational mathematical modeling, network analysis, and have developed an enormous number of tools, and I'll talk just about a couple of these. Ilya Smulovich is one of the major architects computationally of the Cancer Atlas project, and he's developed some incredible techniques for taking all of this genomic data and extracting it from it interesting kinds of correlations. And I'll just say, for example, some of those correlations have been beautifully used to be able to stratify ovarian cancer into highly malignant, metastatic, poor survival from the rest of the ovarian cancers. So clearly a different type than the more benign forms, and they'll need different diagnostic reagents. Nathan Price is actually developing the ability to integrate metabolic networks against protein-protein networks and gene regulatory networks, where metabolic networks represent a biochemical ground truth, and he's demonstrated they can actually correct and extend the other networks in very, very powerful ways. And when you integrate these together, again, you begin to identify key nodal points that one can think about as therapeutic targets. Nitin Baliga was the first ever to develop a complete transcriptional model of a microorganism and show from that model he could predict 80% of the time correct transcriptional responses to environmental stimuli never before seen by that organism. And he's used this ability to make predictions about networks and moved it into cancer and has some very exciting integrative efforts focused on, again, what are going to be key strategic target points in manipulating cancer. And a lot of them aren't at all the classic signal transduction molecules that we all know and love. So I close by saying systems medicine has reached a real tipping point and is really beginning to change the practice of healthcare now. It through the dynamics, provides fundamental mechanisms and insights. Family genome sequencing gives us very powerful ways for the identification of disease genes. We think we've changed blood into a window that can distinguish health from disease. I think we can stratify diseases effectively into their different subtypes. And I think equally important for drug discovery and application is the ability to stratify patients into their distinct related groups in a way that evidence-based medicine misses completely when you put together 30,000 patients and give them drugs and placebos and extract their features from the individuals, knowing that their genetics and environment are quite different from one another. So you have to look at the individual patients and stratify on that basis. The multi-organ approach, recently we analyzed 100 patients with Alzheimer's and 100 matched controls, and we looked not only at the brain organ-specific blood proteins, but at the liver organ-specific blood proteins, and showed that the liver was quite profoundly changed in the Alzheimer's patients. So that means any disease is almost certainly not going to be uni-organ disease, but it'll be a multi-organ disease. And as I said earlier, that opens up really interesting questions about diagnosis and therapy. I've talked very sketchily about new approaches to drug and drug target discovery. I can say using supercomputers, Elliot Samulovich has done some spectacular things with drug repositioning, using both standardized examples to show it worked, and is now exploring new kinds of possibilities. We haven't gotten into these large-scale, multi-parameter, longitudinal Framingham-like studies, but we're looking at preterm birth with 2,200 patients, at cardiovascular disease with 7,000 patients, and we're just beginning a wellness study of more than 100,000 patients. And I think it's these longitudinal studies that are going to create unique opportunities for understanding the earliest and most fundamental aspects of disease in the future. So, I hope I've left you with a sense that whether it's diagnosis or stratification or identification of drug targets, or even thinking about how to make better drugs, systems biology has something to say. Thank you.
Okay, so while we're waiting for people to come up, so I remember when I was a physician, one of the biggest problems was distinguishing a viral from a bacterial infection. It seems like a very trivial problem, and it seems ideally suited to systems biology approaches. Do you know if that's been fixed?
I don't know that it's been fixed. I've got one. But I think it is absolutely a straightforward kind of application that could be done now. We have colleagues at Seattle Biomed who are interested in infectious disease. We're exploring some of those kinds of things.
And then the other thing is, if I had a nodule in my lung, I wouldn't be 100% reassured by an 8% probability that I had a malignant tumor.
You know, the important point is you just wait in watchful waiting, and then and there's a very low probability you have anything. After 3 months, you do a CT scan. If it's unchanged, then the probability goes up into the 90%. Okay. That's point number one. And point number two is we think we can enormously increase sensitivity and specificity just by additional samples and things like that. And we've already started to do that. So we hope to be able to get to a 98% call of malignancy and a 70 or 80% call of the benign nodules.
Okay. All right. Thank you. If there's no questions, we'll move on. Thanks very much.
Okay.